All right, welcome everyone. This explainer is built just for you, the international pharmacy graduates getting ready for the PEBC evaluating exam. Today, we're going to dive deep into the predictive science behind how drugs actually work in the human body.
So here's the big question, right? A patient takes their medicine, and then what? What happens next? The beautiful thing is, the answer isn't guesswork. Nope, it's a science. It's a science built on core principles that let us forecast a drug's entire journey. Okay, let's get into it. To predict that journey, we really lean on two big interconnected ideas. You can think of biopharmaceutics as the starting line. It's all about the drug itself and its delivery system. Then you have pharmacokinetics, ADME, and that's the race itself. It's what the body does to that drug once it's on its way.
So here's our game plan for today. We're going to follow the drug's path, step by step, starting with the foundational laws of biopharmaceutics. From there, we'll trace it through each phase of ADME: absorption, distribution, metabolism, and elimination. And then we'll wrap it all up by seeing how all this knowledge comes together to help us design really smart, effective dosing for our patients.
Okay, first step, biopharmaceutics. And you got to remember, these aren't just guidelines or suggestions. They are the fundamental laws of chemistry and physics that control the very first steps of a drug's action in the body. It all boils down to just a few core properties. These principles, things like how well a drug dissolves, the size of its particles, and the basic laws of diffusion, they literally determine if a drug can even begin its journey.
Okay, this concept is super straightforward, but absolutely critical. A drug has to dissolve to be absorbed, period. And the speed of that process is directly linked to particle size. It's simple physics. Smaller particles mean more surface area, which leads to the drug dissolving faster and ultimately getting to work faster for the patient.
All right, so the drug is dissolved. Now what? How does it actually get across membranes, like the wall of your gut? Well, for most drugs, it's a process called passive diffusion, and it's all governed by Fick's law. All this really means is that drugs naturally move from an area where there's a lot of them to an area where there's less of them. The bigger that difference, the steeper that gradient, the faster they move. And this table here, it's a perfect real-world example of Fick's law. Think about a transdermal patch. If you pack more drug into that patch, you increase that concentration gradient we just talked about, which pushes more drug across the skin. The drug also needs that perfect balance of being able to dissolve in both fat and water. We call that the partition coefficient, to navigate all the different layers of the skin. This isn't just theory, it's the science that makes these products actually work. Okay, so now that we've got those physical laws down, let's see them in action in the first stage of ADME, absorption. This is where it gets really cool because it's where those basic principles meet pharmaceutical technology to control exactly how and how quickly a drug gets into the body. Just look at this. We've got an incredible variety of dosage forms we can use. A simple tablet, a capsule, those little enteric-coated beads. Each one of these is a sophisticated piece of engineering. They're specifically designed to release the drug based on the very principles of solubility and diffusion we just covered. And this digoxin data, it really drives the point home. Check out the 2-hour mark. The oral solution gives a blood concentration that's 6 times higher than the tablet. Why is that? It's simple. The drug in the solution is already dissolved. It gets to skip that whole dissolution step that the tablet has to go through first. So, how do we track this whole journey? We use this. This is basically our map. It's called the plasma concentration time curve, and it is the single most important tool we have in pharmacokinetics. The up and down axis shows how much drug is in the blood, and the side-to-side axis shows the passage of time. Right, so the drug is absorbed, it's in the bloodstream, but it doesn't just hang out there. Nope, it starts its journey within the body. And this brings us to the D for distribution and the M for metabolism. Let's go back to our map here. See that initial upward climb? That slope represents absorption winning the race against elimination. The drug is coming in faster than it's going out. The very top of that curve, that's what we call Cmax, the maximum concentration. And the time it takes to reach that peak, that's Tmax. All right, what goes in must come out, right? That brings us to the final phase of the journey, elimination. This is the E in ADME, and it covers both metabolism and excretion. It's the body's exit strategy. Okay, now we're looking at the downward slope on our curve. This part of the map represents the elimination phase. At this point, the body's processes for clearing the drug are outpacing any leftover absorption, so the overall concentration in the plasma starts to fall. Now, a really key concept for your exams is how this elimination actually happens. Most drugs follow what we call first-order kinetics. Think of it like a funnel. The more drug there is, the faster it gets eliminated. So a constant percentage is removed over time. But a few drugs, like alcohol, are different. They follow zero-order kinetics. This is more like a conveyor belt moving at a constant speed. A set amount is removed over time, no matter how much is there. And that brings us to one of the most useful, most practical numbers we have in pharmacokinetics, the half-life. It's exactly what it sounds like, the time it takes for the drug concentration in the body to drop by half. This one number is incredibly powerful. It's our main guide for figuring out how often a patient needs to take their medication. And here's where it all comes together, the ultimate goal of all this theory, applying it in a clinical setting. How do we take all this predictive power and use it to design a dosage plan that's both safe and effective for a real person? So we use a really systematic, kinetic approach. It's not guesswork. Step 1, we look at the drug's known ADME profile. Step 2, we define its therapeutic window. Step 3, we do the math to design a dosing regimen. And this is crucial, it doesn't stop there. We always have to monitor the patient and be ready to adjust. And this is what we're aiming for, the therapeutic window. You can think of it as the sweet spot. It's the range where the drug concentration is high enough to work, that's above the minimum effective concentration, or MEC, but still low enough to be safe. Our entire goal is to keep the drug's plasma level floating right inside this window. So what happens when we give doses over and over again? Well, the drug starts to build up, or accumulate. This happens because we're giving the next dose before the last one has been fully eliminated. And if you keep doing this, eventually the drug level reaches this plateau, what we call a steady state. That's the point where the amount of drug going in with each dose equals the amount of drug being cleared out. Theophylline is a classic, perfect clinical example of why this is so important. It has what we call a narrow therapeutic window. That means the line between an effective dose and a toxic dose is really, really thin. For an asthma patient, if their level drops below 10, they could have an attack. If it goes above 20, they face serious toxicity. That is exactly why around-the-clock dosing is so critical for drugs like this. It's all about keeping those drug level ups and downs safely inside that target window. So, we've seen how these two fields, biopharmaceutics and pharmacokinetics, give us some pretty incredible predictive power. And that leaves us with a final thought to chew on. As our understanding gets even better, how can we use this data to move beyond standard dosing? To truly personalize medicine for every single patient, taking into account their unique genetics, their kidney and liver function, their lifestyle. That right there is the future of our profession.